Okay,
Yeah.
well, maybe we'll get started because we are recording. So anyone who joins later can always hear what they missed on the recording. If you don't mind just muting for a bit, we definitely will have a Q&A session at the end, and you can unmute or you can put questions into the chat. So, hi everybody, welcome to our webinar. My name is Jana Ullman. I'm one of the founders of the IQSEC2 Research and Advocacy Foundation. I have two children. My daughter Rachel will be 19 years old in December. She has an IQSEC2 mutation. We first realized that Rachel was not meeting her developmental milestones during the first year of her development. And at that time, she was given an autism diagnosis and a Rett syndrome diagnosis. But then in 2014, when she was almost 8 years old, we did a full exome sequencing and discovered that the root cause of her issues is an IQSEC2 mutation. And that's when I started that IQSEC2 mutation Facebook group. So that's where most of you know me from. And we have over 1,200 members now in that group, which is really quite amazing. I will be moderating today's discussion, and I want to briefly introduce Dr. Andrew Levy and share a little bit about his background, and then I will turn it over to him to give us an update on the research that his lab is doing. So, first, I'm really excited to introduce Dr. Levy. He received his bachelor's degree from Yale University and his medical degree and PhD from Johns Hopkins University. He also did his postdoctoral training at Harvard Medical School here in Massachusetts, where I live. And he's a full tenured professor at the Technion Israel Institute of Technology Medical School in Haifa, Israel. He's published approximately 200 peer-reviewed articles in research journals, and his work is supported by peer-reviewed grants from national and international sources. Dr. Levy is also the father of 5 children and 17 grandchildren, with his second grandson having an IQSEC2 mutation. And that is what led him to move his lab over to study IQSEC2 disease about 6 years ago. So currently, his lab is entirely focused on trying to find a way to improve the lives of children with IQSEC2 mutations. And he is honored to work with the IQSEC2 Foundation and the parents of children with IQSEC2 related disease. And we are extraordinarily honored and thrilled to have him working for our shared cause here to improve the lives of all those living with IQSEC2 related disease. So that is a very quick bio, but I know everyone is very eager to hear his update on the research he's working on. And again, we will have time for Q&A after his update. So with that, I'll turn it over to you, Dr. Levy, whenever you're ready.
Thank you very much. Really, it's humbling to present to all of you. And I wear two hats. One is as a parent, grandparent, and the other as a scientist. I'm going to break this presentation up into two parts. The first part is to make sure that everyone can understand at a very, very simplistic level what we're talking about when we talk about gene therapy for IQSEC2. So for some of you, this will be very, very simple. For others, it won't be. I tried to make it, though, as simple as possible. As mentioned, this will be recorded and you can review it. I want to make it clear that if anyone has any questions, I get, many emails asking questions about the research and things. You're always free to contact me. Anything that I say today to clarify, be my pleasure. So again, we're going to talk at a very basic level, a very simple level for about maybe 20, 25 minutes. And then we're going to move to questions, which some of the questions I've received already. Some of them are quite detailed or at a high scientific level. So for some of you, that might be over your head, but I thought that was a good way to break up the presentation and our discussion for today. I have as much time as needed. So anybody who wants to stay on and ask more questions, it's not a problem. Okay. All right, so I'm going to break up this first part of the talk today, talking about these four ideas as we talk about what is gene therapy, how we're going to go from having the mutation in IQSEC2 to the therapy with a new gene. First, we're going to talk about the mutations. All of you know, of course, that you have a child with an IQ mutation. We'll talk about what is known about the different mutations. And then we're going to talk for the other three parts of the talk as I get to it, and we'll follow this general outline. Okay. So the gene, as most of you know, is on the X chromosome. There's two copies of the gene in females, one copy in males, because boys only have one X chromosome. The basic dogma when we talk about in molecular biology is that we go from DNA to RNA to protein. So if we imagine that this is the portion of the X chromosome, a very small portion of the X chromosome, which is the IQSEC2 gene, we can think of it as being a long string of beads, which we refer to as nucleotides, and there are 4,464 bits of information or nucleotides in the gene. Each of you have a child with a change in one of those nucleotides, one of those beads on that string of 4,400 nucleotides. The DNA is then converted into something called RNA for the IQSEC gene, and then it is turned into protein. It is translated into protein, and it has 1,488 amino acids. Again, shown here as little beads on a string. And each of you, with your son or daughter, has a mutation in one of those beads. We'll talk about more of the different types of mutation in a few minutes. This string of beads then folds into a specific conformation referred to as the IQSEC2 protein, which functions using a variety of functions, which we really won't go into too much today, but I'm happy to answer questions about, and we published quite a bit on that as well. What's known about the different types of mutations is there are at least 150 different types of pathological IQSEC mutations. What does that mean? It means that if we look at all the children that have mutations that result in disease due to IQSEC2, 150 different examples of the mutation was in a different bead or different nucleotide of the IQSEC2 gene. That's an important feature in terms of what types of therapy might be amenable. We'll come back to that perhaps in the question and answer session. About 75% of the mutations in our children create what is known as a premature stop signal. When you have a gene, the RNA determines that specific beads or amino acids are incorporated into the protein. In some of us, in some of the children, though, there is a mutation which encodes a change in the DNA, which results in there being what we refer to as a stop codon. This results in a premature stopping of the protein synthesis. So a shorter protein is made than is normally made. Again, about 75% of the children have such a mutation. This type of mutation results, this results in the RNA for the IQSEC2 gene being destroyed, so no protein is then made. The other 25% of the children have a protein being made, we call a missense protein, in which one of the amino acids has been exchanged for another amino acid, one of the beads in that string of amino acids in the protein. This results in an abnormal protein, which may lose its function or gain a new one or become overactive. So essentially, we can think of all the mutations in our children as falling into two functional classifications. One is class 1, in which no protein is made, no IQSEC protein is made due to a premature stop codon. you can look at your genetic report and you can identify that that's occurred by there being an asterisk after the specific amino acid that was mutated in your child. But again, about 75% of the kids have this. The other two, 25% of the children have an abnormal IQ protein, and we're calling that class 2 mutations. What determines the severity of the mutation and the response to therapy? It's not possible still to say for a specific mutation whether that mutation is going to have a worse prognosis or a more moderate prognosis. What we do though know is that by gender, in general, boys have a much more severe disease than girls. This is probably because the girls, of course, have a normal X chromosome in addition to having a mutated copy of the IQSEC gene. And we know that among girls, there's a tremendous variability in the severity of the disease. This is probably due to what we call skewing of expression from the two different alleles or two different forms of the gene in girls. We don't know necessarily that the class 1 and class 2 are worse, one or the other. There are certain types of class 2 mutations, though, that appear where the protein is working overtime that appear to be even more severe in terms of their manifestations and the disease that it presents as. So let's talk about how we model the disease and how we are developing precise therapies to treat IQSEC disease. One approach that we're using in modeling the IQSEC disease is to use what's called induced pluripotent stem cells. What we're doing here is we take a small biopsy from the child and we culture these in a skin biopsy, and we culture these in a petri dish, and we generate a culture of skin, what we call skin cells or skin fibroblasts. We then genetically reprogram those fibroblasts and turn them into what's referred to as stem cells. Stem cells, you may have heard about, have the ability to be then differentiated or turned into any cell type that you want. A heart cell, a liver cell. In our case, we turn them into brain cells or neurons. We then can study the neurons in a petri dish, and we can assess how excitable the cells are, which is the equivalent basically of a seizure. We can also determine how the cells speak to one another, sending signals to one another, which is a measurement of what we refer to as synaptic function. The importance here is that we can study the disease in a petri dish from any child with the same genetic background as the child with the IQSEC2 disease. So, it's a problem sometimes with doing things in animals or in other model species because the genetic background, other than the IQSEC mutation, may be different. But here we're using the exact same genetic background as the child has. And we've initiated this with quite a few children, and we're expanding this dramatically to be able to look at a large number of children using this platform. The second model that we're using is using mice. And what we're able to do is we're able to genetically alter the DNA of the mouse to produce the same IQSEC mutation that's present in any child. This is using, by and large, a technology referred to as CRISPR. You've probably heard about in the news, but we can pinpoint any of the spots in the mouse IQSEC gene and turn it into the same mutation that occurs in a human case. What's interesting is that the mouse and human IQSEC genes are virtually identical. The amino acid level, it's about 98% identical. So this allows us to make the same mutations in the mice that occurs in human. We can then study the class 1, the knockout, in other words, there's no IQSEC protein being made by those mice, or the class 2 mutations in which there may be a change in function of the IQSEC2 protein. What we've been able to demonstrate over the last couple of years, these mice models recapitulate The seizures, in other words, these mice have seizures just like the children have. They have abnormal learning abilities, and they have behavioral abnormalities in terms of their ability to communicate with one another and their ability to socialize with one another. So how do we use these animals to then approach gene therapy for IQSEC2 disease? And I'll present to you now a proof of concept study in animals that we've just published in a peer-reviewed article. I know that many of you through the Facebook page have gotten access to this paper. I hope within the next day or two, this will also appear on the Foundation website, as well as a simple explanation of the paper going into some of the experimental details. I'm also going to talk about that right now, but the paper itself should be available to all of you. It's also available online. You can also obtain it, and I think or you can get it through the Foundation in the next day or two. Okay, so let's talk about what we've done so far in gene therapy for IQSEC mutations. We're going to focus specifically today on knockout mutations. Again, these are the children. We're modeling a disease in which no IQSEC2 protein is made. We've done now a proof of concept, meaning we've been able to demonstrate that we can treat and rescue the disease present in mice with IQSEC2 knockout mutations. These are, again, about 75% of the kids. Using gene therapy, we can correct the abnormalities in their seizures and their behavioral abnormalities using this gene therapy approach in the mice. Basically, the strategy, given that there's no IQSEC protein being made, is simply to replace the absent IQSEC2 protein. We're going to talk about in a minute how we're assessing the efficacy of the gene therapy, the kind of tests that we're doing to show that it actually is helping the mice with this same mutation that's occurring in our children. And I think an important question for many of you is, when is the therapeutic time window for gene therapy? In other words, when is it possible to give this and there still to be benefit? Okay. We're using as the platform for our gene therapy a type of virus called the adeno-associated virus, or AAV. This is a virus that occurs naturally in nature, and it doesn't cause disease in people. The virus itself cannot reproduce itself, unlike most of the viruses that we're used to thinking about, but it does have the ability to infect human cells, and it can exactly exist in the cell for years, essentially a lifetime, without ever coming and integrating into the actual DNA. It just exists as a little circular piece of DNA outside of the human genome, your chromosomes. Each virus has its own, I'm using a term I don't know if you're all familiar with, ways or navigation system to get to a particular part of the body and cell type. For example, the AAV type 9, and there's well over 50 different types of AAVs that have been identified, is specific for targeting the cells in the brain called neurons. This specificity or this targeting is determined by the outside covering of the virus called its capsid, or which particular cells the virus will be going to or be navigating towards. So if we look inside the virus, you see the outside is the capsid. The inside of the virus is its DNA. The DNA gives instructions to build new virus, to replicate, to allow the virus to function, essentially. What we do, though, is we take out the viral DNA, the normal viral DNA of the adeno-associated virus, and we can replace it with any gene that we want. In this case, we replace the AAV genome, the normal genes present in the AAV, with the IQSEC2 gene. This allows us, using the capsid, to direct the IQSEC2 gene to neurons, and then it allows the virus to deliver the IQSEC gene to the neurons in this case. How do we design this for treatment of IQSEC2 mutations? There are a couple of constraints that are important. The total amount of DNA that can fit into that AAV that I showed a picture a minute ago, this particular structure, can only fit in approximately 5,000 nucleotides. Optimally, even a little bit less than that, about 4,700 nucleotides. So we have some constraints that make it more challenging because the IQSEC2 gene is quite large. Itself is close to 5,000 nucleotides. So we need to find a way to decrease the size of the IQSEC gene. We're using functional genomics and AI to do this. And we also need to find ways to regulate the IQSEC gene so it is only turned on in neurons. Even though the virus only goes to neurons, it can still potentially infect other cells in the brain. And the gene is only turned on, though, using specific signals we incorporate into the virus only in brain cells or neurons. How do we assess the efficacy of the gene therapy? Again, using the induced pluripotent stem cells turned into neurons, we can essentially test that the virus can prevent seizures in a petri dish and restore synaptic function. This would allow us to determine if the therapy we develop in the mice, for example, works in a given child's mutation, given his or her genetic background. A disadvantage, of course, is that this is in a petri dish, and we can't look at cognitive behavior. We can't look at behavior, can't look at other types of behavior as well. So now I'm going to talk about the data in the published paper that IQSEC2 knockout mice, these again, mice not making any IQSEC protein, we can rescue them. We can treat these mice with replacement gene therapy. So again, these mice have been genetically engineered to make no IQSEC2 protein. We then have created a adenovirus type 9 with the human IQSEC2 gene using a neuron-specific promoter with a genome size that is a little bit over what is optimal, but it still allows the incorporation of the DNA without it being truncated, about 4.9 kb. Cooperating with several groups around the world, one in Japan and one in the United States, and of course in our lab. These two other laboratories have replicated and demonstrated that we can correct the social behavioral abnormalities, the abnormal seizure susceptibility, and the impaired growth in the mice that have a IQSEC2 knockout mutation. I want to emphasize the importance of these two groups worked independently, of course, of our laboratory with their own IQSEC2 knockout models. So this is an independent replication of the work, which is very important in this type of work in science to increase confidence that we indeed are able to have a verifiable result. So I want to talk about some of the tests that were done in this paper. You can look at the paper itself, but just to introduce the concepts to you so you can understand what we're talking about. Again, we're talking about replication independent labs and in a knockout mutation. So two types of tests that we were doing, looking at behavioral abnormalities as well as looking at memory. The first is referred to as social preference. Mice are just like people, very social animals. You would prefer probably to talk to another person as opposed to talking to an object, although maybe there are some people that would like to do that. But in general, we prefer to talk to other people, just like mice would prefer to interact with other mice as opposed to an object. So this is on the left-hand part of the slide. Mice with IQSEC2 mutations show no social preference. They show no preference for an object as opposed to another mouse. When we treat with this IQSEC2, human IQSEC2 in the knockout mice, we're able to restore the social preference of the mice to now prefer associating with another mice as opposed to an object. The second type of looking at behavioral, in this case even memory, is doing what's called stranger preference. In this case, we have the mice are exposed, in first case, to, this is our test mice here. He's exposed to another mice of the same type, referred to this as the familiar mouse. So he's now familiar with this particular mouse. He associates with him. And now we then put him in a cage with both of these mice, and we see which of the mice he would prefer to go to. As I said, mice prefer novelty. They like to go to new things, just like people, rather than going to something that's necessarily old. Mice that have a mutation in their IQSEC2 gene would do not have any preference. They go to equally these two mice. Mice in which the IQSEC2 gene has been restored now prefer to go to the stranger mouse, the new mouse in the cage. So this is a type of behavioral abnormality, not preferring novelty, not preferring a mouse over an object, which can be rescued in these mice. These are classical tests looking at a variety of gene therapies that are used to assess the ability to restore behavioral abnormalities for other similar types of disease, like Rett syndrome and other diseases as well. Another type of abnormality that we were looking at was simply the growth of these mice. As is true of many of your children, I know some of the children grow and weight quite normally, but for many of the children, they have difficulty with feeding abnormalities. And in the mice themselves, this is the case that there's an impaired growth, particularly in the neonatal period, and that is, in other words, equivalent to your children up to about age 2 or 3. The mice, and shown in the lower panel, that get the virus tend to grow very similar to the normal mice. They gain weight at the similar rate as normal mice. Mice that don't get the virus have a stunted growth and don't gain weight as quickly as mice that do get the virus. The third type of test that we're looking at is seizures. Mice with a knockout mutation have seizures, like other mutations. And many of the children, of course, as you know, have seizures. We are able to monitor seizures by looking at EEGs in the mice. And we can also assess the threshold for developing a seizure in the mice, again, using a type of EEG and test as described in the paper. Mice with a knockout of IQSEC2 have an altered threshold for seizures. In other words, their ability to have seizures is different from normal mice. We are able to restore the threshold to normal when we give back the IQSEC gene. This is showing a normal threshold, and this is an abnormal threshold in a mice that has not received the IQSEC gene inside the AAV9. In addition, because these seizures can be quite severe in these mice, the administration of the virus actually improves survival of the mice that get the virus as opposed to mice that don't get the virus. Several limitations of our study that we published. The amount of the protein that is existing in the cells after we administer this virus is not identical to what exists in normal mice. We are not controlling the protein expression levels to be identical to what's occurring in the normal mouse. A second limitation, we're not seeing any toxicity in the knockout mice in terms of any abnormal behaviors, or certainly in terms of growth or in survival, but it may impair growth in normal mice. We also, as you'll see in the paper, we administered the virus to normal mice, which of course we would never do to a normal person. But in those mice that already had a working IQSEC2, too much of the protein was actually appeared to cause an impaired growth or a stunted growth. Finally, the size of the viral genome is still suboptimal. It's approximately 4.9 kb instead of 4.7 kb. And we believe we found a workaround for this, but this is another potential limitation. I alluded to this earlier. When is the therapeutic window? What does our data show regarding that? We're extremely pleased to find that even outside of the neonatal period, even in other words, even when the mice are in young adulthood, which I would say the equivalent would be in their maybe up until the early teen years. In a mouse, if you can draw that analogy, but the mice are certainly now sexually mature. The mice are still able to be rescued for these behavioral abnormalities. This gives us tremendous hope that not only for the very young children, but for the older children, we will be able to hopefully be able to provide benefit. This is not particularly so surprising given the tremendous synaptic plasticity that occurs in children up to their early teens, but it's not the case for all gene replacement strategies. So we think this is a very important finding. Now, what about class 2 mutations? I spent the entire time talking about the class 1 knockout mutations, 75% of the kids. The class 2 mutations, we're taking a different approach to be able to do this. So we currently have not been able to demonstrate in mice yet to be able to rescue or cure the type 2 mutations, but we believe that we should have an answer within a very short period of time. The strategy we're using here is very similar to what I described for the knockout mutations. We're providing a therapeutic IQSEC2 gene, and we're providing a specific way to make sure it's only expressed in neurons, but we're using another component. In this case, what's referred to here is the shRNA. This is a piece that we've incorporated into the virus, which essentially turns off the mutant. Turns off the, please silence yourself or mute yourself.
Bleib ruhig, Lia. Ich habe jetzt hier ein anderes Problem. Ich bin hier rausgefallen.
Tanya, if you can mute your phone. Thanks.
Okay. Tanya.
So these short hairpin, the SH stands for short hairpin RNA, is able to specifically degrade the overfunctioning or abnormal IQSEC2 RNA, and then we replace it with a normal IQSEC gene. That's shown in this particular slide. So we're going to, in the class 2 children, we're trying to do a simultaneous, what we refer to as knockdown or elimination of the abnormal gene, and then replace it with a normal IQSEC gene. That's shown here. So this is the virus we're seeing here. It gets into the neuron. The shRNA specifically attacks the mutant RNA and targets it for destruction. The therapeutic IQSEC gene that produces a normal IQSEC2 protein. In this way, we can then, we believe we can treat the class 2 children in this manner. We've been able to get around an important problem as we're doing this by miniaturizing the therapeutic gene to get down to a genome size of under 4.7 kb that will allow this strategy, we believe, to be tested and hopefully to be successful in the coming weeks, months. So having presented to you basically our approach, our data for IQSEC2 knockout mutations and our work towards trying to treat the class 2 children mutations, I'm going to talk now briefly about moving from preclinical models to clinical trials. Where are we right now? We've demonstrated benefits in animal models by independent laboratories, referred to as preclinical validation, that we have a gene therapy approach that can treat knockout mutations for IQSEC2. The next step, which is currently undergoing, this is being evaluated by independent experts. This work has been published, as I said, under peer review. The work is also going to be presented at the American Epilepsy Society meeting in December. We're assembling another panel of independent experts to go over all the data, both regards to safety as well as the preclinical data, to get their opinions regarding the next step in moving towards a clinical trial. Moving towards a clinical trial will involve several important features. First and most importantly is making sure that the procedure is done safely, and this will require that it goes through ethical approvals, data submitted regarding the preclinical data, as well as evidence from other studies with very similar types of viruses that have been used in children with similar types of diseases that we hope will allow this to pass all the necessary ethical approval requirements. And this will be done in collaboration with, again, with physicians from around the world and with hospitals that are going to be actually carrying out this study. What you're all, I'm sure, very interested in is when will the trial be launched? This depends on many factors. Certainly the ethical approval, I think, could easily take up to 6 months, maybe up to a year to get the approval from the necessary authorities. Funding for this is now being initiated in a small number of children. We're anticipating the study should be done in about maybe 5 boys with knockout mutations. I think no more than 5 in this first study, and they will receive, this is a one-time treatment of the virus. Child will probably be hospitalized for up to a week, no more than that. The entire treatment lasts approximately 10 minutes, in which the virus is administered as one bolus directly into a compartment in the brain called the ventricle through a procedure which is frequently done in children with a variety of types of cranial disorders. It's a safe procedure. It's being done for many other syndromes, including Rett syndrome, this approach for how the virus is administered. Again, we anticipate this will be done in approximately 5 boys with knockout mutations in this first clinical trial. We then anticipate that the children will be followed looking at, obviously, at seizures and a battery of tests looking for developmental milestones such as speech and other types of progression over the next 6 to 12 months to assess the efficacy of the treatment. The goal certainly here is to make this available to all children. All children, we want this to be available to, regardless of what the mutation is going to be. But the first study is going to need to be done in children with boys with knockout mutations where we do have evidence that this is efficacious. And we'll talk in the next part of the talk about what's going on with girls and with other types of mutations. But in this first study, this is the optimal population. We believe that if we can demonstrate in these 5 kids benefit, this will open the door in terms of getting additional support for a larger study and more children, opening this up to the entire community. This will also help in getting insurance companies to pay for whatever treatment costs will be associated with this. And I think this is the appropriate approach to do this. I'm sure there'll be questions about this in the question and answer session. What I'm going to move now to is to talk about some questions. Some of you may have already have these questions. These were sent in earlier to the foundation, and I'm just going to briefly talk about these questions about the work and about the potential trial. The first set of questions have to do with treatment considerations in girls. And the question was, are there specific differences for treating girls with IQSEC mutations, given the X-linked inheritance and X inactivation? One important feature that differentiates IQSEC2 from other genes on the X chromosome is that it is not X inactivated. Its expression is generally skewed towards one of the 2 X chromosomes, one of the 2 alleles. If it's towards the good allele, it's child has a mild disease. If it's bad allele, child can have very severe disease. The concern here with the current approach that we're taking with simply replacement strategy is that the girls already have a normal copy of IQSEC2. And given that we are not currently controlling levels of IQSEC2, this may require a knockdown of the current IQSEC2 gene in the girls, and then a replacement, similar to what we've approached in the class 2 individuals, the class 2 children. So at the present time, we don't believe we have evidence that this is effective in the girls. This will require much more extensive work. There's problems in the mice model. The mice IQSEC gene is very different in terms of its X inactivation than the human IQSEC gene. So we're limited in the kind of the extrapolation from mice models with IQSEC2, what we can do in with the mice, but we can do this in the pluripotent stem cells and the differentiated neurons in the girls' cells. What active research is underway specifically for girls right now? What stage is it at? Again, we're doing preclinical research with the pluripotent stem cells, pre-programmed fibroblast cells from the girls, turning them into neurons. We can then study the different viruses that we develop in the lab for gene therapy and determine if this can eliminate their seizures in a petri dish, as well as their ability to communicate with one another. This is going to require additional work, but we're actively engaged in this and collaborating with several other laboratories in this. Have you explored female-tailored approaches, X inactivation related strategies beyond the current AAV gene addition? As I said, the IQSEC gene is not X inactivated, so what's done for some genes that are X inactivated in girls is that one approach is to try to reactivate the inactive, what may be the healthy gene. In this case, that's not really on the table, given that the gene is not inactivated to begin with. Could the skewed expression be eliminated? It's a very interesting question, and again, will require more extensive research. Next question. What's known, and some of these questions, as you appreciate, are fairly technical, but again, I'm happy to field all questions about anything. What's known about the roles of the short and ultra short isoforms in pathology and development? Basically, there are 3 different isoforms of IQSEC2 that are known. There's one that's about 73 amino acids. There's one that's 949 amino acids. There's one that's 1,488 amino acids. The function of the very short form is totally unknown. Even the existence of the protein has not been demonstrated. It shares no function overlap with the intermediate or larger forms. The intermediate form is expressed outside of the brain predominantly. The major isoform that is expressed in the brain is the large isoform, the 1,488 form. This long isoform is the major isoform in the brain. It's responsible for synaptic activity due to the presence of a specific domain which allows the localization of the protein to the synapse. The other forms of IQSEC2 gene isoforms do not have this ability to be localized to the synapse. Furthermore, we are able to rescue the knockout mice only with the large isoform, the long isoform. Taken together, all this data suggests that the major isoform that we should replace and we are focusing on is the large isoform IQSEC2 gene. Does your current construct restore all relevant isoforms? No, it only restores the long isoform. How important is isoform balance for efficacy and safety? Is this being measured in your preclinical work? The importance of the isoform balance requires further study. As I mentioned before, we're able to rescue simply with the long isoform. It's quite complicated to study the isoform regulation, of course, in the brain of their children. When looking, it is possible to study the isoform regulation in cells outside the brain, but of course, what's going on in cells outside the brain, for example, in monocytes and macrophages, has been shown to not necessarily correlate what's going inside the brain. So this is a very, very good question that one of the parents asked about this, but a lot of these questions require further research, but I think that we feel very comfortable that the focus here is on the long isoform of IQSEC2. Do IQSEC2 mutations often arise at conception or later in development? virtually all the mutations are thought to arise very early in development. There have been a few mosaics identified. In other words, the mother actually that has several oocytes that may harbor the gene. This has been reported in a very few number of cases. Virtually all the cases, well over 95% of cases, are de novo arising early in development. How will mosaicism be handled in trial design? I think what the question here is not referring to mosaicism in the mother, in other words, that she can give rise to several children with the same mutation. But actually in some children, the mutation occurs later in development. So the child can have some cells that are normal and some cells that are actually mutated. I think the answer to this question really in the strategy we're going to be taking for children with mosaicism, and it's similar concerns to what's going on in the girls. We want to avoid overexpressing IQSEC2 in cells that are already expressing IQSEC2 in the normal cells and these children with mosaicism, which we anticipate is a relatively small percentage. I'm aware of 1 or 2 cases like this. And certainly these will not be in the initial study. Do you expect mosaicism to influence the therapy response and safe monitoring? Well, certainly, I think, again, the attack we're going to be taking is very similar to what we need to take in girls who are essentially a type of mosaic as well. Are you in contact with the Rett syndrome community to share learnings on female X-linked neurodevelopmental disorders? Actually, our overall strategy we're taking is very, very similar to what the Rett Society is using in their current phase 3 clinical trial with gene therapy. Of course, the Rett gene is very different from the IQSEC gene in terms of its function and the approach that's taken in terms of the design of the vector, but the same, very similar type of strategy, the way that the vector is being designed, the same type of virus, the AAV9, administered through the same route is what's being done for Rett therapy now in phase 3 clinical trials, and it has shown efficacy in some children. And actually in one child that were receiving very high dose, there was a very severe outcome. Actually, one child died in that particular Rett study. Are you collaborating with international groups working on gene editing or other gene modifying technologies that could be relevant to IQSEC2? So, you know, we're a relatively small group with the funding that we have. We are focusing on using adeno-associated virus to try to treat IQSEC disease. The CRISPR-based strategies are progressing very rapidly. The current hurdles are what we refer to as off-target. CRISPR can correct mutations, but it can also create new mutations, referred to as off-target effects. The efficiency of the correction is also reduced in cells that are not dividing, which is the case for neurons in the brain. And the delivery to the neurons is much more difficult than is the case, for example, to the liver or to blood cells, which is where this is really being used right now. The field is progressing very rapidly. This is has always been part of my thinking, the strategy that we can take to more quickly develop treatments that can help our children. This is not the end of the story, what I presented to you today, gene therapy using an adeno-associated virus. It very well may be that within the next couple of years, a CRISPR-developed strategy for editing mutations due to IQSEC2 will be developed. But this is what is available today. And we believe we'll be able to go into clinical trials within the next 1 to 2 years for this using this approach. One additional thing I'd like to say about CRISPR, and I think this is true for all CRISPR-related diseases, is that you would need a different virus or how you're going to deliver the CRISPR enzyme to do the gene editing for every single mutation. In this case, we have 150 different mutations. This will require a different, if you will, drug for each of the children. This is not prohibitive, but this would dramatically increase costs. What we're proposing here is that for the 75% of the kids with the knockout mutation, all these kids can receive the same virus. This should reduce the costs and the timing of being able to develop a treatment for our children. A single drug or reagent can be used for all of the children. Is gene therapy useful for all mutations within the IQSEC gene? I read that gene has to be reduced. This, again, we may require different approaches. I've alluded to that today. The class 1 and class 2 mutations appear to require a different approach, but again, a similar gene therapy approach that we are working on. We believe for the class 1 mutations, we have a viable candidate for using now in children, and we're working on developing one for the class 2 mutations. Does the response to therapy depend on the protein where it is truncated? And yes, of course, that's what we focus on today. For the truncated protein, we're going to be using the replacement approach, simply a normal copy of the IQSEC gene delivered by the virus. And okay, that's for my presentation, and I want to open it up to all of you for any questions you may have about anything that I spoke about or anything that I didn't speak about.
Thanks so much, Dr. Levy, for driving this research, for telling us about it, for explaining it in a way that we can understand. There are a few questions in the chat, and also thank you to folks who submitted their questions ahead of time, which we've already gone through. Most of them are clarification questions, so maybe we'll start with those, Dr. Levy, if that sounds okay to you. One clarification, is the knockout mutation the same as a missense mutation, which I know many people see in their report?
No. A knockout mutation specifically means there is no protein being made for IQSEC2, okay? It is due to a premature stop codon or termination. If you look at your child's genetic report, he or she will have an asterisk next to where the mutation is. If anyone who wants to send it to me, I can tell you whether or not you have a stop mutation or not. But usually in the report, it also says this is due to a premature stop codon. A missense mutation is when there is a change in one of the 1,488 amino acids. So the entire protein exists. There is IQSEC2 protein, but this results in a change in the function of the protein. It may be totally dysfunctional, or in some cases, it may actually cause the protein to work overtime and be always doing its function, which itself is a problem. In other cases, it can actually cause the IQSEC protein to add a new function, what we refer to as a gain of function, that itself could be toxic. So missense and knockout mutations are very different. What we've talked about today, the therapy is specifically for knockout mutations or nonsense nonsense mutations.
Thanks for that clarification. The next question I know you touched on, but we've gotten many questions about this, so it might be worth just repeating once again, but are there plans to conduct tests involving girls?
Yes. Okay, so I think first of all, obviously, we're not going to do any tests in girls before we have evidence, preclinical evidence that whatever treatment we're doing is efficacious, ethically and just not responsible to do. So we believe that the disease as presented to DA is different in girls than it is in boys, predominantly because of the genetics, again, because it's an X chromosome. So what we're doing right now in the girls is we have fibroblasts, we are induced pluripotent stem cells, turning these into neurons. We're able to assess if we're able to rescue the abnormalities in the girls with these particular viruses. This is a large grant that we've applied for with a group from Germany that has approximately 40 children from Germany with IQSEC mutations. And we're going to be obtaining the induced pluripotent stem cells from all these children. A high percentage of them are girls. And we'll be able to assess some of the viruses that we've developed, either the virus I talked about today or the class 2 virus that I described a little bit using the knockdown and replacement approach. So I see this progressing in the human cells. That's the avenue we can test this in. The problem in the mice is difficult. We certainly will try this in female mice as well, but the disease and the genetics is different for IQSEC2 in female mice than it is in female humans. It's not the case with in males. There's a single copy, but there is no such thing as in the mice. There is X inactivation of one of the alleles, but does not occur for the human case for IQSEC2. So it makes it more difficult to extrapolate the mice model in girls from mice to humans.
Thanks for clarifying, Dr. Levy. Dr. Levy, do you want to stop sharing your screen just so we can see you a little bit more?
Sure.
That's great. Thank you.
Okay.
A few more questions in the chat that we'll cover. The next one I'm going to interpret a little bit, but I think the question is the therapy that you're thinking about currently a one-time administering, or is it something that will have to be done ongoing? And tell me, Mira, if I'm misinterpreting your question, but I think that's it.
This is a one-time treatment, referred to as a one-off treatment. As I mentioned, it takes about 10 minutes to administer the virus as a single bolus through a catheter. What we know from studies where you can actually look at the tissue afterwards, this is done in monkeys generally, as much as 10 to 20 years later, still see the virus still present. We anticipate this should be able to work for the lifetime of the child, a single treatment. How long will it take to actually work and see efficacy? This is an excellent question. We anticipate certainly over the 6 to 12 months where we are certainly anticipating seeing a change in the seizure profile if the virus is effective. With regards to learning and behavioral abnormalities, this is a more difficult question. How quickly the child will regain or relearn or learn new abilities. I like to think about it this way. If we think about neurodevelopment as a upward slope, all of you have many of you have other children. You can see that they develop milestones at given age at 15 months, at 2 years, what they're able to learn. And your child with an IQSEC2 mutation has referred to as a more shallow slope in terms of their progression. They will not necessarily get to some of the milestones that your other child has developed, but also whatever they do learn, they learn at a slower rate. I see this as being able to increase the slope or the rate at which they learn. Obviously, the next day they're not going to start have these skills at hand, but it will allow them to start learning. I think that's probably the most important feature here. It will allow them to start learning using intensive therapies and be able, hopefully, to make up lost ground that they had from other children. This will be, of course, why the earlier we intervene, we anticipate the greatest benefit will be in those children that are the youngest when we administer therapy. But as I alluded to in the talk, we still believe that it will be beneficial even giving into older children, even in young adults, we believe.
Thanks, Dr. Levy. We are at the cusp of a really exciting time. There's a lot to do here. I keep saying it's a marathon, not a sprint, where a lot is changing. An incredible amount has changed in the last 10 years, and we're really on the cusp of the next major shift. Next two questions I think are the same, which is really more details on how the selection process will go for these trials, on who can participate.
Right. Of course, I understand the angst regarding this. And first of all, I will not be involved in making this selection. This will be done by an independent review board of clinicians and scientists looking at the data and most importantly, at the profile of the children that potentially could be involved. I think it's fair to say that it makes the most sense to start with children that we have the most chance for success and showing the most benefit. This will benefit the entire community if we can demonstrate benefit in these first few kids. We want to start with a child in which can demonstrate the most benefit, and I think that will be in children that are at the youngest age possible, whether this, usually the disease manifests sometime around age 1 or 2. And so probably children of that age will be the children that will be actually enrolled, the first children enrolled in the study. That makes the most sense, in my opinion. But again, that will be a decision made by an independent board We want to make completely open as to how that process will be made. And that board will go through the data. It's all children. Then, of course, any parent that wants to participate can submit their child as being potentially for the trial. But I think the first trial will be done probably in the youngest children as possible. As far as for the stem cell research, a similar question. Here, really, there is not as much of a limitation. It's more a limitation on what our laboratory can process. We're hopeful that we obtain this international grant with the German group. We'll be able to open this up to virtually the entire community. But we'll know more on that probably in November or December. Okay.
There's also people have some specific questions about their own children's mutation. Maybe what is the best way for them to
Just send it to me by
send
email.
email. So I'll put your email address into the chat at the end, but I'll skip over any sort of specifics about individual children. I don't think we should do those over the call, over the recorded Zoom call. So next question, is there a tentative timeline for the clinical trials and where will they take place?
So there actually is likely this is going to probably be done in Europe at one site. More to come, where exactly which site that will be. We are going to be developing there is a clinical research organization which is going to be developing and helping with all the paperwork that will need to be submitted for this particular study. There's actually a small company that's being formed by one of the parents. I let her identify herself if she wants to. Otherwise, keep that anonymous. But she is taken on to start a new company. I'll let her to talk about it if she would like to in a moment.
Hello everyone. I will let you know a little bit about the company in a while.
Thanks, Vasia.
Vasia is a mother of a child, a young boy with a IQSEC2 mutation. She's graciously started a very small company focused only on IQSEC2 to try to get this clinical trial to be done in a small number of children. I think it is realistic that we will be able to get the approvals for doing the study within the year, and I think hopefully within 6 months. I think the actual mechanics of getting the necessary funding and getting the study, the hospital on board, getting all the other regulatory issues involved, makes it likely that this could take up to 2 years before the first time we can get the virus into a child. I'd like this to happen within the next 6 months, but we have to do this safely. We have to go according to all regulatory issues that's required by law, and that's what's going to be done. But again, the timeline that I presented to you is how I see things progressing. I think there's a lot of excitement, a lot of momentum for this. Some of it is not entirely in our hands, again, with the regulatory agencies when they review things like that. But that's what I would say. Certainly within 2 years is feasible, and hopefully earlier.
Maybe this is a good segue to talk about funding. There is a variety of different sources of funding that Dr. Levy is looking at. Vasia is working very hard with this company to raise funds. You've heard me talk on the Facebook group where 100% of the money we're raising through the IQSEC2 Research and Advocacy Foundation, we are putting towards these clinical trials as well. So we're trying to look at a variety of sources for that funding. Obviously, it is quite expensive to go through this process and make sure it's effective and safe and following all of the right procedures that Dr. Levy talked about. So I just want to put a plug in for participating. Our strollathon is something we've done every year where people all over the world, where we're all located, we picked this weekend, September 26th this year, to try to fundraise locally with your networks. I'm going to put into the chat our fundraising page. And, again, whatever you can do with your networks to help support the clinical trials. We're all the foundation is 100% volunteer. We don't take any money from it. It all goes to the to support these scientific research efforts. So just want to put a plug in there. There are a few more questions. One other question about the age range. So someone with a daughter who is 24 years old, how long might it be before she might be able to access treatment?
Okay. We don't know the age limit on this. I said there are clearly, the brain is amazing in terms of what we refer to as plasticity, its ability to respond to injury, for example. You see, for example, after people have had strokes, even in their 50s or 60s, they can regain function. Some people can, and some people do not. That reflects a degree of plasticity. Other parts of the brain are taking over for other regions that were injured. I think the same thing is true here. I think that we don't know when the window, so to speak, is, if it is closed at a certain period of time. We have done some studies in referred to as adult mice, which may be relevant for your daughter. The separate issue, of course, in girls versus boys, but will this type of therapy be appropriate? I think it is fair to say that I think it's something that if this therapy shows efficacy, so in other words, it works and provides benefit, it is reasonable that it would be tried regardless of age, with the caveat that the net benefit will probably be less than in a child, almost certainly be less than a child that was 2 or 3, but still benefit, particularly on things like seizures, if that's a problem for your daughter, and potentially even in achieving milestones. I've seen with other genetic diseases where therapies are started at a later age, it's still possible to, we like to talk about in terms of milestones, in terms of incremental benefit. It's still possible to achieve new skills even at a later age. That's how I would look at this. So I would be optimistic that if this shows benefit in the younger children, that we will try it in all children and young adults.
I like to think of it that there will be some treatment in my daughter who's 19 years old or almost 19 years old now. There will be some treatment that will make her life easier. I feel really confident that that will happen, and I really look forward to that. Vasya, would you like to give a quick
Yes.
update on what we're putting you on the spot. We just talked about this in advance, but we'd love to hear from you.
First of all, I would like to thank Dr. Levy for explaining everything today to all of us and for carrying our hopes and dreams for our kids all of these years. And Zana as well for creating this community of parents, which is very important to take for the next step that we're going to take. I want to thank all the parents that I've met, and they've asked how we are going to take this forward, how can we help? Because this is in a few months, it's going to be on our hands. It's not that we're going to be asking when is this treatment be available, but what can I do to make this treatment available? So from my part, since we did not find the big companies interested in helping us create the therapy for our rare disease, I took the chance. I'm not in the biotech business. I'm an educator. We have a family-owned private school in Greece. It's out of my knowledge what I'm doing right now, but it is important that I took this step for my child and for our group. We created a business in biotech in Austria with the help of another biotech company here in Greece who has shown us the way. In order to be able soon enough, after we solve some legal issues, to be able to buy the patent and proceed with the funding and create a roadmap in order to get to the clinical trials. I hope it's someone else, but if it's me, I will take the chance and try to help lead this way. What is the purpose of this company? Since a big investment is going to be needed for the clinical trials, we will try to attract funding either from angels or from the industry or through the funding of different foundations of parents around the world, so that we have those 5 kids doing the clinical trials. The next step, as we see it, is going to be persuading all the companies that will, not the companies, the companies that will be able to give us the treatment for free. For example, for each country, it's going to be different approach and a different way to see to show that the treatment is less expensive than the treatments that the governments are now paying for our kids. So different foundations that are popping up around the world are going to be playing a very important role. It was one in Italy that was created in the beginning, then the one that we created based in the States. There's a German one now, there's a Spanish one, and it's really important to have this going on in all over the world. So I'm feeling that we are on the same, on the right track. It's important for many people to get involved in this because everybody's providing solutions, and we're moving even closer to our goal. And I wholeheartedly trust the work that Dr. Levy is doing, and thank him. Without his research, would be nowhere right now. So that's it from myself. Thank you.
Thanks so much. Thanks so much, Vasia. And it takes an international village to help keep moving this forward. So thank you so much. So just maybe 2 other questions from the chat, and then we'll wrap up, and we will definitely share this recording. One question around the timeline for the missense mutation treatment. So not the knockout, but the other class, Dr. Levy, that you were talking about. Is there a rough timeline there?
Right. So we have mice models of the missense mutation, which reproduce the effect seen in children with specific missense mutations. And we are now testing. We have in our pipeline approximately 120 different virus constructs that we've created, and we are testing them simultaneously by members of our laboratory. And we have some that look promising, but I think hopefully I'll have another update when we find something that actually that provides benefit in this particular model. I'm hopeful, but science goes in not predictable ways, but I'm optimistic that we will find a solution for this in the coming months.
That is great. And then one last question. I've had many people ask me this over the years, but is there value in coming to the United States for treatment? From this question comes from Pakistan.
No. For the gene therapy, for example, with this company that we described, this is probably going to be done in Europe. Treatment in general, I don't think so. It's important though that you have access to medical services. I think through the Facebook group, you can talk with other parents to make sure that you have access to the medications that are available to children in other countries. I don't know what the medical reimbursement situation is there as well for making sure your child has access to whatever is necessary for controlling his epilepsy or her epilepsy if he has it. So I don't see a value necessarily in coming to the States, no. why that would be necessary at this point.
Great. Okay, I think that's it. If you have questions specific to your child, there were a couple of those in the chat that I skipped over, but I did put Dr. Levy's email address, since he so kindly offered to take further questions over email. This session was recorded. Hopefully, Dr. Levy's office will cut out our banter at the beginning and just have the relevant portion. And please feel free to share this webinar, share these updates. Whatever you can do to help us with fundraising, obviously, is incredibly appreciated. And that's it. So hopefully we'll meet again when there's further updates, and we'll keep you posted via the Facebook group. Thank you. Thanks, everyone. Thank you so much, Dr. Levy, as always.
Thank you, everyone.
Thank all of you as well. I'm really privileged. I look forward to speaking again to all of you soon. But again, please don't hesitate. Contact me by email if I'll try to answer as quickly as I can.
Thank you.
Thank you, Andy.
So
Thank
much.
you.
All right. Bye, everybody.
Thank you.
Bye, everyone.
Thank you.
Thank you.
Bye.
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 Merci.
Thank you.